Buenos dias. Good morning and bonjour. So my name is Stephane Boissel. I'm the chairman and CEO of SparingVision. And it's my real pleasure to be back in Barcelona, I think 2 years after the last in-presence conference in the same place. So what is SparingVision? SparingVision is a genomic medicine company which is 100% focused in the ocular space, in ophthalmology. And you can see on the slide a summary of the story of the company. The company was started in 2016, but the biology of the lead asset that we have in a portfolio today really started way before, actually in the 90s, but it's in 2004 that the team of the scientific founders really identified RDCVF, which is short for rod-derived cone variability factor, as the main hypothesis beyond the cone preservation in the context of RP, retinitis pigmentosa, or other inherited retinal disease. Anyway, it took 15 years for the founders and the academic team to really validate the hypothesis, to decipher the mechanism of action of the biology, and then in the mid-2010s, with the growing incompetence when it comes to gene therapy, they've decided to put the biology, and I'm oversimplifying, to put the biology into an AAV, and they decided to start SparingVision. In between 2016 and 2020, the company was more or less managed as an extension of the academic lab it was coming from. This is the Institut La Vision in Paris. And in 2020, the company was a single asset company with a lot of holes in the racket, like I like to say, and no management team. And this is when I was lucky enough to be appointed CEO of the company. I was already chairman of the board. I moved back from San Francisco, where I was working with Mark from Sangamo, and was appointed CEO on top of my chairman role. And from there, with the board, with the incoming Series A investor, we've decided to really accelerate the story, build a real management team, and also start to expand the portfolio, as we were not very comfortable with such a novel biology to develop a single asset company. 2021, a few months after this change, we had a very busy year when it comes to corporate development. First, we acquired another company, another spin-off from the Institut La Vision called Gamut. And that acquisition gave us access to two new products that I'm going to speak to in a very short moment. And very importantly as well, in 2021, we were very happy to meet the team of Laura from Intelia and enter into a strategic relationship with Intelia, which, as you all know, is the world leader in vivo genome editing. So a very busy year in 2021. Now we are in 2022, and we are a few weeks away, a few months away from submitting our first clinical trial application, first in Europe, that will be in July, and then in the US through an IND process in September with our lead compound, SPVN06, which is, in short, the RDCVF biology. I think it was important for me to give you the backstory for you to better understand what will come next. So we have a new team. I think we have one member of the team in the room, Dan Sheng, and Dan has been with us for 12, 13 months. And we are very fortunate to have Dan on board, because Dan was previously at Spark and previously at Shop and UPenn, and he has been following the Luxturna story, which, as you know, was the first gene therapy product approved in the US, in the eye, on top of that. And he has been following this story from really the bench, the mice experiment, to the market launch. And Dan, again, thank you for having joined the team on the venture a few months ago. And the rest of the team is composed, as you can see. I'm sure some of the names will be familiar to you, from real experts in our field. We've raised 60 million euro in Series A with prominent investors in the like of Jeto, UPMC, Isios Capital, which is headquartered in Barcelona, Foundation Fighting Blindness, FFB, BPI France, the French sovereign bank, and 4Bio. And then when we acquired Gamut, we paid the acquisition in shares, so we welcome AdBio, which was previously Advent France as a shareholder. And then when we did enter into the strategic alliance with Intelia, we paid the upfront for the license in shares, and so Intelia became a shareholder of the company as well. Now, what is SparingVision about? So SparingVision is about, again, a genomic medicine play in ophthalmology. And we develop our portfolio across 3 different pillars. The first pillar is a pillar composed of gene therapy product. But unlike anything else being done in the space, all our gene therapy products are gene independent or mutation agnostic. And on top of that, they are disease independent. And I will come back to what does that mean in a short moment. So this is where the legacy asset is coming from, SPVN06, the one that should enter into in-man testing in 2022. And this is where also the Gamut product that we acquired in 2021 are fitting in. In the middle, the second pillar is a pillar that is composed or will be composed of the product we will be developing jointly with Intelia. And here we are going to go into mutation-specific product based on the CRISPR toolbox that Intelia will be bringing to the collaboration. And then the last pillar on the right of the screen, which has a different color code. It has a different color code because we have yet to start anything concrete, and that will be very likely what we will do when we will be closing the Series B. It's a pillar in which we want to develop in vivo reprogramming product. I'm sure you're familiar with what is currently being done with the likes of iPSC technology to ex vivo, try to turn certain cells into other cell type. Here, we are going to raise the bar a little, as we will try to do with transcription factor attached to AAV, LNP, or whatever vector. We are going to try to do what is being done ex vivo, but in vivo. So the bar is going to be very high. So it's fair to say, if you project yourself in the next 4 to 5 years, this is what we are doing as we are raising our Series B, it's fair to expect a lot of value to come from the first pillar on the left. Some value, I hope, as much as we can from the pillar in the middle, but don't expect anything from the last pillar as we have yet to start anything. And this is more for what will come after tomorrow. This is the portfolio presented in a more traditional way. Again, we started the journey like 18 months ago with one single product, SPVN06, and we have now pro forma a portfolio of 6 products and still counting. The reality is that with the Series A, we are actively working on 3 products, SPVN06, SPVN20, and the first of the Intelia target product. SPVN06, as you will see, it's an AAV, but the cargo inside the AAV is what we call a neurotrophic factor or neurotrophic cargo, which aims at working on the metabolism of the cone so as to preserve the cones from degenerating. So we are not, unlike a Luxturna, unlike, again, anything that you can see in the space, we are not trying to restore function, restore vision, but we are trying to preserve what is left of the vision at the time of intervention. And this is very important for you to understand. SPVN20, and by the way, sorry, very importantly, I said it at the beginning, but I really would like you to take that away. This product is expected to work independently of the causative mutation of the disease. Okay, and I will come back on the reason why if I have time, but time is running. SPVN20, the second product, the lead product that we acquired from Gamut, is again a gene-independent or mutation-agnostic product, but unlike SPVN06, the AAV will have a different cargo, a cargo which is a GIRC transgene, which will aim at restoring function, but not in a normal cone, or in a cone that has some defect, but a cone that has lost its ability to function as part of the evolution of the disease. Those cones, are called dormant cones. They are cones that have lost their outer segment, and as you probably know if you know the space, this is where the outer segment, phototransduction is usually happening. So the idea is to inject GIRC into the inner segment of the cone so as to mimic in the inner segment of the cone what was previously, when the cone was normal, done in the outer segment of the cone. And I won't be able to speak to the Intelia target because this is something we are going to keep secret as long as possible for obvious reasons. We have 2 important presentations coming. As you know, ARVO is a big show when it comes to the ophthalmology space. It's in 2 weeks' time. We are lucky enough to have an oral presentation and a few posters. The oral presentation will be on the prospective natural history study. I cannot say much today, but we have learned a lot, and we have learned a lot of things that will be super useful in this natural history study for the upcoming phase 1 trial. And then at AGCT, we've been selected for an oral presentation on the preclinical development, preclinical data, and preclinical journey of SPVN-06, again, the lead asset. I'm going to skip this slide just to tell you that we are advised by the best of breeds in our space. And I'm not going to spend too much time since, again, time is running, but I just want you to take away when it comes to the gene therapy portfolio, this slide. This slide is a schematic of the evolution of RP, retinitis pigmentosa, but this is a pattern that is valid across most of the inherited retinal disease. And this is where or how we are going to position the product. SPVN-06 is going to be positioned at a time where patients have lost their rods. It's a time when most of the patients are diagnosed. Actually, 80% of more of the patients are not diagnosed when they are born, but they are diagnosed in their 20s, in their 30s, when they start to bump into things. They start to see less, at night or in a less lighty environment. And the reason why they are in this state is because they have lost most of their rods. And the rods are the photoreceptor that you are using at night. But to look at the screen today, you are using your cones, okay? So we are going to intervene with SPVN-06 at a time where most of the rods are gone, and we are going to substitute with our product, SPVN-06, the rods in their function to supply the cones with this neuroprotective factor that I was referring to before, okay? SPVN-20 will come at a later stage, same disease, same pattern, except that we will target, as I was saying earlier, dormant cones which are viable, but which have lost their ability to function. And SPVN-30, a product for which we have IP today, but we have yet to start anything concrete, will be, in a way, a broad spectrum of action in which we will be combining the transgene from the first two products in what we expect will be, again, addressing a very, very large population. I'm speaking way too much, sorry. Importantly, as a takeaway message, when it comes to the expected clinical outcome, on the left side of the slide, you have what to expect with a Luxturna or with any vision restoration or function restoration product. You expect to change the course of the disease by restoring some function. With SPVN-06, our goal is to slow down or to stop the progression of the disease. And bear in mind, this is a very slowly progressive disease. People will lose vision in 20, 30, or 30 years from diagnostic, okay? So if you can just change, the course of the disease by a few degrees, you will enable people to gain maybe a decade, maybe 2 decades of vision. I don't have time to go through the biology, so I will go to what we have observed in preclinical model. And as you can see from this sketch on the left, it's a mice model called RD10, a very aggressive model in which the mice are losing total vision in 2 to 3 months. And we've been able to, at 3 different time points, to see a clear difference between the eyes of the mice that have received our product versus the eyes of the mice that have received the vehicle. As of today, we are in the final stage of preparing for IND and CTS submission. We expect, as I said, CTS submission for SPVN-06 in July and then IND in September. So we are at the very last moment of what is called a GLP tox study in monkeys, and so far, so good. So I have all reason to believe today that we will be able to pass those milestones successfully. Importantly, on SPVN-06, very often we receive feedback from investors that the eyes is a very interesting field to target genomic medicine because it's small, because it's easily accessible, because you don't need a lot of product to see something. But at the end of the day, there is a drawback to that. It's very small. These are very small indications. Well, this slide is going to tell you exactly the opposite. When you develop a mutation-specific therapy, such as Luxturna, yes, indeed, the market will be very small because you are going to target only a tiny fraction of the market. Being mutation agnostic, where Luxturna in Europe and the US will probably address a target population of 3,000 patients, we will be targeting 350,000 patients. So if the biology works, and even though we cannot expect the same pricing as a Luxturna, Luxturna was priced $425,000 in the US per eye, even if we take half of that price, the market potential for this product is enormous. There is no equivalent to the market potential of this product anywhere in our space. Okay, and that's the beauty of being gene agnostic or mutation independent. And same goes for SPVN20. I'm only left with 30 minutes, so unfortunately I don't have a lot of time to go through the rest of the presentation. But just to summarize what is coming next for SparingVision. Again, a super important year, 2022, first-in-man study. We expect to enroll the first patient in December, Dan. I hope we will be able to make that milestone because your bonus depends on this one. We expect with SPVN20 to be IND ready in the first quarter of 2024. With a different mechanism of action, we expect the two products to read out clinically more or less at the same time in 2025. It will be slower for SPVN06. It will be shorter for SPVN20 as we are looking at restoring function. We expect multiple preclinical catalysts, from the Intellia partnership in the next 4 years. We expect to continue to expand the pipeline. I mentioned in vivo programming, but we are also looking at in-licensing or buying what we call enabling technologies to do life cycle management on the portfolio. And last but not least, we are going to grow significantly the headcount. There are 35 of us today, both in the US and in Europe, and we expect to grow to 70 to 80 at the end of 2023. And we will invest in process development infrastructure as well as possibly in GMP manufacturing. Sorry, I had to go super fast, but I really thank you for your attention today, and I wish you a very good day, very good conference. Thank you.